Good afternoon. My name is Matt Cartmill, and I'm here to welcome you on behalf of the Boston University Department of Anthropology to the first of a series of experiments in new conferencing procedures called the Boston University Dialogues in Biological Anthropology. These dialogues are intended to bring attention in the BU community to current issues within the field and to make them accessible to BU faculty and students. And more importantly, they're meant to bring together well-known biological anthropologists who share a mastery of a common literature and an interest in the issues at hand, but hold divergent views on the topic. And our objective in staging and broadcasting and organizing these dialogues has been and will be to provide a model of productive scientific discourse, academic discourse, for BU students that will serve as something of a counterweight to the usual sort of media talking head event in which two people with irreconcilable opinions on a topic yell at each other for 10 minutes and then cut to a commercial. We'd like you to take away from this experience today the proposition that it's possible for people who disagree about something to talk to each other in ways that actually affect the way they think and affect the things that they know. So today's dialogue is going to be about the curious case of the little woman from the Indonesian island of Flores. About 140 years ago, when Charles Darwin wrote his big book on human evolution, The Descent of Man, very little was known about the fossil record of human evolution. There was practically nothing to build on, and so Darwin was able to speculate that human evolution started with something like a modern ape and ended up with something like a modern human, and that the intermediate stages were all just sort of filling in the segue from one stage to the other. But as we've learned more and more about the fossil record, it's become evident that the human family tree has branches on it, and that there have been times and places when more than one kind of human or human ancestor lived. And exactly when and where these times and places were are still a matter of debate. Practically everybody in the field agrees that the early stages of the human evolutionary story had a lot of branches in them, back before 2 million years ago in Africa. But exactly what was happening between that time and the present in our own genus, Homo, of which we're the genus Homo sapiens, is still a matter of active and intense scholarly debate. Sometimes new discoveries have caused us to subtract branches that we thought were on the tree, as in the notorious case of the Piltdown hoax, which was unmasked in 1950 and caused us to simply rub out that whole branch on our tree diagrams. Other discoveries have caused us to add branches. One of the most recent of these, and one of the most controversial and hotly debated, is the little female or woman that was discovered in 2002 on the Indonesian island of Flores, and which has been reported as variously as a new species of the genus Homo, surviving until astonishingly late in the history of our genus, and also has been interpreted alternatively as an individual with some pathologies that make them look a lot more different from modern humans than they actually were. So we've brought together two people here today that have rather different opinions about some of these issues. And we're going to begin with Dr. Fred Smith, from who is chairman of the Department of Sociology and Anthropology at Illinois State University, and the outgoing past president of the American Association of Physical Anthropologists, a distinguished and prolific author and researcher about the later phases of human evolution. So, Fred, I'm turning it over to you.
Thank you, Matt. First of all, I'd like to say it's a great pleasure to be able to participate in these dialogues. I think this is an excellent mechanism to get discussions out on important issues in the discipline of biological anthropology. I want to make two sort of general statements before I start. First of all, as a human paleontologist, I'm usually very, very reticent to make any presentation on fossil material that I myself have not studied. So I have to be direct up front and say that I have studied directly none of the fossil material from Flores. And so what I'm going to present is entirely from the literature. It's not from my own research, and I think it's important to let that be known up front. In the same regard, I have no horse in this race, so that I have no vested interest in any of the particular interpretations of this material that may be out there. So again, thanks Matt and Kay for the invitation to participate, and we'll get started. I'm going to present some general information on the enigma of the Liangbua people, because I think they do represent an enigma. And so first of all, let's talk about what we have at Liangbua. A human skeletal sample of 97 elements. 61 of those elements come from one individual, the Liangbua 1 partial skeleton, 14 from another partial skeleton, Liangbua number 6. No other individual has more than 3 elements. So there are 9 to 14 individuals represented, but again, only 2 of them are relatively complete. The others are in fact represented perhaps by only 1 skeletal element. The dating from the site is from approximately 95,000 years ago to 17 to 16,000 years ago, so very recent in time. There are 2 major periods of human occupation that have been defined, one between 75 and 61,000 years ago, and another between 18 and 17,000 years ago. And these are located in different parts of the cave system. So the Liangbua 1 partial skeleton and the Liangbua 6 skeleton are from units 7 and 11 that are close to the east wall of the cave. The earlier time period is more towards the center and the western wall of the cave, and this is a picture of the Liangbua 1 partial skeleton. Some of the other facts. There is an interesting faunal association with the Liangbua humans that include Komodo dragons, large rats, large birds, some of those predatory, and a pygmy form of elephant in the genus Stegodon. There are also a large sample of stone tools, including rather well-made blade tools, and the humans and the stegodons appear to go extinct around 17,000 years ago because there is a massive volcanic eruption that is visible in the sequence at that time. And after this time, the stegodons and the Liangbua people are gone. And then you have more recent, clearly modern people on the island at about 11,000 years ago. So here's where Flores is located. It's part of the Lesser Sundas in what is now Indonesia. It's important to realize, however, that Flores is to the east of Wallace's line, and so therefore never connected to the Sunda Shelf. And anything that got to Flores would have had to at least cross some open water, even at time periods of very, very high or low sea level. In addition to the location of the Flores site, the site of Liangbua, which is indicated by the skull here, there is another area on the island that is significant for our discussions. This is the Soa Basin, which is located a bit to the west, a bit to the east. The Soa Basin has a couple of sites that have tools, no hominins, no human fossil material, but tools. The most recent one is the site of Wolosege, which has just been published in Nature. And from this site, we also have a nice series of stone tools. Here are some of these. Perhaps not quite as nice, some of them, as the subsequent tools that are much later in time at the Liangbua site, but certainly suggesting that humans were on the island of Flores roughly around 1 million years ago, because the date at Wolosege is about 1.02 million years. Okay, there are essentially 3 interpretations of the origin of the little people of Liangbua. One is, and this was the original one, that they represent an insular, a dwarfed variety of Homo erectus. We know Homo erectus is in the area somewhere over 1 million years ago, and so these individuals at Flores represent insular dwarfs that that represent a group that underwent this process, which is a relatively well-known process among mammals, from presumably large-bodied ancestors that got onto the island. I could have changed this to 1 million years ago. The second interpretation is that these are basically modern people, Australo-Melanesian pygmoid people, and that Liangbua 1 has some combination of pathological conditions that result in its rather unusual morphology. And probably the most recent interpretation, which came out a year after the initial dwarfed Homo erectus interpretation, is that the Liangbua people represent a part of an even earlier radiation of Homo out of Africa prior to the radiation of Homo erectus. So, here is a diagram by Bill Jungers and Karen Bob that show the two possibilities, at least as far as an evolutionary explanation from earlier forms of human, the dwarfed erectus form, and then the derivation from an even earlier Homo form. What is interesting is that if, again, as we said, Flores was separate from the Sunda area, the currents are very strong in this area. So it's thought that when the humans got there, they must have probably come from the north, not just across the Lombok Strait, but probably got to the island from somewhere to the north. And the general idea is that something like a scenario or something like a tsunami carried a large group of these people onto the island. This established the population that then developed on the island in total isolation for upwards of 1 million years, resulting in the Liangbua people that we are talking about today. Okay, I'm going to start by just making a few comments about the Liangbua postcranial material. There are two specimens where we have a lot of postcranial information. They give us heights of about 1 to 1.1 meters. The Liangbua 1 individual that you see here has a very short femur, femur that is at the very lowest range of any modern humans, and it has limb proportions that show, relative to the femur, longer arms. And the limb proportions that this individual shows are out of the range of modern humans. The humerofemoral index that Dr. Bill Jungers has reported recently is 86.8. This falls out of the range of all moderns, including pygmies. And here is some data from Dr. Jungers' work in which he basically points out that the limb proportions here are very similar to earlier, more primitive forms of humans, such as Australopithecus afarensis, the famous Lucy fossil, and so forth. But again, even if you look at small-bodied living populations, African pygmies, Andaman Islanders, and so forth, they do not, even though they have smaller body sizes, that does not involve the change in limb proportions that we see in the Liangbua individual. If you look at the iliac blades in the pelvis flare out beyond the acetabular rim, and this is a condition that we don't see in any other modern humans, including very small-bodied humans, but we do see it in earlier forms, again, such as these Australopithecines. And also, the joint for the hip, which is reflected in the acetabulum of the os coxa and the femoral head, and the femoral head here is shown here in comparison to the femoral head from Lucy. The femoral head is very small, which again falls out of the size range of modern humans. So if we go to other parts of the skeleton, the clavicle, the bone of the shoulder, is very strongly curved and it's very short. There's a low torsion angle of the humerus. In other words, the humerus is not twisted on its head like it is in modern humans. And this reflects the condition that you see in earlier humans, but again, not in small-bodied modern humans. There are also primitive structures of the wrist, which I'm really not going to talk about at all here. And there are also relatively elongated and potentially some primitive features of the feet compared to lower limb size, and there is some suggestion that there might be some weight-bearing functions to a bone called the navicular, which normally in modern humans is not a weight-bearing bone. So, sorry, that's repeating. Whoops. Here we go. So here is the comparison of the foot bones to the tibia. Again, this comes from Bill Junger's work at Stony Brook University, and particularly the middle bones and the terminal bones of the feet, the toes and the actual foot bones themselves, are elongated. The lateral toes, other than the first toe, seem to be a bit curved. These are all relatively primitive features, and the important thing to understand is that if you look at small-bodied modern humans, they do not show these kinds of features. They don't seem to be an aspect of just reducing body size. The Liangbua skull is extremely interesting, and actually most of the controversy surrounds the skull. Has a very small cranial capacity, has a primitive vault shape. It has some primitive features such as anterior pillar-like structures along the side of the nose, which you see mostly in Australopithecines, some Australopithecines, and particularly it has no chin structures. It has a receding mandibular symphysis, has a primitive internal symphysis, which looks very much like earlier humans, particularly Australopithecines. And here I'm just showing you a comparison of a single microcephalic skull to show you that there are some similarities here, but certainly these conditions are not identical. And I want to particularly, emphasize that in the microcephalics that I've seen directly or have in the literature, all have well-developed chin structures. So even in this specimen up at the upper right, you can see the chin structure. If you look at the overall shape of the cranial vault, the overall shape of the cranial vault seems to group with primitive humans and not to group with microcephalics. The microcephalics are in the bottom right part of this diagram on the left side of the page, and the Liangbua specimen is the star that you see here, which is up with the earlier human specimens. The mandibles, as I said, are primitive. They're small. They have primitive features of the symphysis. They may have, if you look at, microcephalics, they may appear to have a negative chin, but when you look at the bone structure, it turns out that the chin is present. It's just the prognathism in the alveolar region where the teeth are covers this up. So again, the pattern that you see in the mandibles you don't normally see in even small bodied individuals today. There are some also primitive features, particularly of the premolars, which you see normally in Australopithecines and early Homo. These primitive features, which I'm not going to go over because I don't have enough time, are found in modern populations, but at a low frequency. And the real crux is to try to figure out what's going on with the brain, and my colleague Bob Martin is going to discuss that, so I'm just going to skip over this. So I want to end by saying that there are three very troubling questions to me. I look at this postcranial material and I think, okay, this is not something that is caused by small body size, and it's probably not caused by any pathology, although comparisons with pathological postcrania have to date not been done. But there are still three things that bother me. Where is the evidence of this pre-erecting radiation from Africa? Well, maybe we have a little of it. Okay. What is the probability that there was only one tsunami that brought people to Flores? I mean, would this have only happened once? And if it happened more than once, what was the impact on other people coming into the Flores population? Were they really isolated for all of that time? And if they weren't isolated, then how do we explain their derivation from an early human radiation? And finally, if you look at the stone tools, although there's certainly some continuity in the stone tools, there's also some progressionism in the sense that some of the later tools do show a little bit greater sophistication. So does this occur in isolation? Is there some innate tendency here to develop better stone tools? I'm not sure. But this bothers me. So I do want to point out that there might be some additional evidence of this pre-erectus radiation from the very recent publication of the DeSilva human mitochondrial DNA genome, which seems to be basically twice as different from modern humans as Neanderthals are. So perhaps this is some evidence of this, but I really think we need a little bit more. And I'll just end by saying, if you look at some other examples from the history of paleoanthropology, we do see examples where the first specimen of what ended up being a new taxonomic group, at least in some cases, was very heavily criticized at first, and in the case of Neanderthals and Rudolf Virchow, pathology was used as an explanation for this. In the case of the Taong child and Australopithecus africanus, it was clearly the issue of, well, this is a small subadult individual. We have to know what adults look like. And more recently with the Tumai Sahelanthropus skull, which is heavily distorted, there are debates as to whether this is an early human or not. And my point by showing all of this is that with the first two examples I showed you, the solution to this question came only with new discoveries, new material, new information, new studies. It probably wouldn't have been solved if those specimens remained the only ones possible. And I think the same thing applies to Sahelanthropus and to the enigma of Liang Bua.
Thank you very much, Fred. I appreciate it. Our other speaker on today's inaugural BU dialogue is Robert D. Martin. Dr. Martin has a long and distinguished career in all sorts of aspects of biological anthropology, including for many years being director of the Anthropological Institute at the University of Zurich in Switzerland. At the moment, Bob is the curator of biological anthropology at the Field Museum of Natural History in Chicago, and he is, among other things, an internationally recognized expert on the evolution and growth of the brain in primates and in humans. So, Bob, take it away.
Thank you very much, Matt. And I'd first of all like to say also that I'm very pleased to be able to participate in this debate, particularly because that's precisely what's been lacking so far, is a debate about this supposed new hominid. And what we really need to do is to get the two sides together and discuss this. And so I really welcome the opportunity to do this. First one I've been offered. The first thing I want to emphasize is that if we compare a modern human skull with the so-called Hobbit skull, the LB1 skull from Langbois cave in Flores, is that it is the only skull. And Fred finished off by saying what we need is more specimens, and we certainly do. There are apparently other individuals based on fragments. The only substantial other individual that we know is a second lower jaw, and all of the others are small fragments. And the impression has been given that this is a population of small-brained hominids. It may be a population of small hominids, but we only know that one individual had a small brain, which is that LB1 individual. The brain is grapefruit-sized. It's a third of the size of our brains. And the thing about that is that struck me immediately, and I really want to emphasize this was my starting point, is that when I saw the first paper published in Nature in 2004, I said this brain size just does not fit. And so if we look at a curve here, what I've done is to put all of the cranial capacities we know from the human fossil record that are fairly reliable, dating back to 3.5 million years on the left of the graph and coming up to modern humans on the right. And you can see that there is a fairly well-defined curve there. And then if we look at the Langbois specimen, LB1, that red point at the bottom shows the cranial capacity of LB1. And it falls right off the curve, and it is the only hominid that does fall right off the curve in that way. And the key point is it's only 18,000 years old. If LB1 were 3.5 million years old, that brain size would not be surprising, or even 2.5 million years old, but it's only 18,000 years old, and that is what doesn't fit. And so various special arguments have been introduced to try to explain that small brain. Many people have simply ignored the problem, but attempts have been made to try to explain why this individual had such a small brain. And one of the first was that this is an effect of what is called island dwarfism, that various mammals tend to get smaller on islands, and so it was suggested that the smaller brain was a result of dwarfing on an island. Well, it doesn't fit the data that we have for mammals generally, and I just want to give a very familiar example to people. We have bred dogs so that they range in body size over a factor of 20 in terms of weight. And so you can get really big dogs and quite small dogs. And this has been through our selective breeding, but it's the same principle. And if you look at the skulls in the bottom illustration, you can see that skull size varies dramatically between these different breeds, but the brain case is not very different between the biggest dog and the smallest. And we can plot this out on a graph. And this is for a score of different domestic dog breeds, so these are averages for each breed, and that is a plot of brain size against body size, and there is a fairly nice linear relationship. And that linear relationship is similar in its details to what we find when we look at any mammal within species to look at the relationship between brain size, body size. In fact, in primates, the relationship is even flatter, so brain size tends to change even less with body size in primates than it does in other mammals. But even if we just take the general mammal figure, the Hobbit would have had to be 1 foot, not 3 feet, but 1 foot high to have a brain that small. Doesn't work. The second special argument that's been used is that we have these exciting new hominids from Diminutive in Georgia, which Fred mentioned, and there are, I think now 5 skulls, but certainly we have cranial capacities for 4 of them, and they're quite small. And so the Diminutive hominids were said to be a possible explanation that a Homo got out of Africa, went through Georgia, and then got to Flores, and that the small brain of the Flores hominid can be explained by the Diminutive hominids. Well, the problem is the Diminutive hominids are 1.7 million years old, not 18,000, they're 1.7 million years old. And if you look at this plot that I showed you before, if we now plot in the 4 Diminutive hominids, those mauve dots in the middle fit the distribution beautifully. They do not fall off the curve. And in fact, the average brain size in Diminutive 1.7 million years ago is 50% higher than in the LB1 skull. So again, this doesn't explain anything. Now, other special cases have been mentioned. One thing I should emphasize is the island rule applies to body size. It's never previously been applied to brain size, and we don't really have much in the way of data. Now, what's been happening is people have been finding special cases and saying, hey, I found an island mammal that has a really small brain. This explains the Hobbit. And one case that's been cited is of a dwarf bovid, a relative of cattle, found on a Mediterranean island called Maya tragus. And it's supposedly descended from another genus of larger bodied form called Rupicapra. And if you look at the skulls down at the bottom, you can see that they look dramatically different. And in fact, Maya tragus, the island form, has tiny eyes. And so clearly its sense organs were very, very different from its supposed relative Rupicapra. If you look on the right, the orange points are the brain size for the island form Maya tragus, and they fall well below the line for other bovids. The argument is that brain size became quite a bit smaller in this island form. Well, the first thing is there is also a dwarf deer that occurs on the same islands, and there is no sign of reduction of brain size in the deer. Second is that we tend to call a small form on an island a dwarf. And this assumes we know the size of the ancestor, that we know that the ancestor was big and it got to the island and became smaller. Now, we don't know this in this case. What we know is that we find Myotragus back to 5 million years ago on these islands. And throughout the time that you find it, it has a tiny brain. And so one alternative possibility is there is a common ancestor of this entire group of bovids that was small and had a small brain size. And that explains why you find this Myotragus form. It's a survivor from a more primitive condition. The other thing is that DNA has been extracted from Myotragus, and it's not related to Rupicapra at all. It's actually related to sheep and goats, which are about the same body size, so there hasn't been any reduction relative to those. Now, the other case that was cited very recently in Madagascar, in Nature, was dwarf hippopotamus, again using the word dwarf, implying that they got smaller. We don't know. What we do know is on Africa we have the standard African hippopotamus, which are the green diamonds up on the right, and that's a relatively big form, and this shows the relationship between brain size and body size. But at the bottom left, there is the pygmy hippopotamus from Liberia, which is also an African form and is actually smaller than the Madagascan hippopotamuses. And in the middle there are two Madagascar hippopotamuses which are only known from subfossil remains. And the key point made in the article was that they seem to have relatively smaller brains compared to their body size than the modern big hippopotamus. But we don't really know when the Madagascar forms diverged from the African big hippopotamus. And if you look at the pygmy hippopotamus down at the left, the brain size difference is less dramatic than if you compare it to the big one. So we have to be very careful in talking about island dwarfism. Quite often you find small-bodied forms on islands. We don't know whether there was a small-bodied ancestor and these are survivors, and the mainland form got bigger, and it's just an assumption that we're talking about island dwarfism. There's another problem with the hippo example, is this kind of treating all islands as equal. Madagascar at the top is 4 times the land area of Java in the middle, and Java is 10 times the land area of Flores. And it's being argued that hippos got smaller on Madagascar and ended up with smaller than expected brains, and this explains the Flores hominid. Homo erectus lived on Java, and the biggest brains recorded from Homo erectus are found in late surviving Homo erectus from Gandong in Java. And so Java, which is 4 times smaller than Madagascar, did not lead to small brain size in Homo erectus. So why should it lead to small brain size on Flores? Now, so far nobody's looked at any primate, and so what we did, this is unpublished work, but we looked at Macaca fascicularis, the long-tailed macaque, which happens to live throughout all of those islands in Southeast Asia. And this is a project conducted by Megan White in my lab. And so what we did was to go to museum collections and check body size and brain size. Now, in fact, there's a very nice recent paper that looked at body size in long-tailed macaques in that region and showed that there is no island effect on body size. Long-tailed macaques do not get smaller on islands. They're just the same size on Flores as they are on in Thailand or anywhere else on the mainland. But nobody had looked at brain size. So here is skull size, and the blue dots are males and the red are females, but in both cases you can see the small islands are on the left and the mainland is on the right, and you can see there's actually a slight trend for a decrease in body size in both males and females. So if anything, the island forms are slightly bigger than the mainland forms. And if we look at cranial capacity, brain size, we find there's no significant trend in these lines. They're just too flat to tell us anything. But again, you can see, in fact, females tend to show a weak trend downwards as you go to the left, and males is weak trend upwards. But there is no significant relationship between brain size and island size in this macaque. It's the only primate that's been looked at so far, and there is no island effect on brain size. So that doesn't work either. Okay, I said my main interest is in brain. It was a tiny grapefruit-sized brain that struck me immediately as a problem. And so I then looked at other things. If we look at the stone tools, these were described by the authors as sophisticated stone tools and in the commentary in Nature. And it's difficult to understand how these could have been made by Homo floresiensis. Several studies have been looking at the postcranial skeleton, the skeleton below the neck. And there have been studies, for example, on the wrist, the foot, the upper forelimb, including the shoulder blade and the thigh bone, the femur. And Fred has already mentioned some of this. And the bottom line is, as in the wrist, if you look at the bottom right diagram, you have chimpanzee first, and then the Hobbit, and then modern humans. And it's clear that this particular bone, the trapezoid in the wrist, looks much more like a chimpanzee than like a modern human. And this is true for all of the rest of the data. If we look here on the femur length, the great apes are in red diamonds, and the green diamonds are humans. And you can see that LB1, which is down at the bottom left, one of the blue squares labeled LB1, actually lies in the great ape distribution and slightly beneath the human distribution. But the other thing you notice is those lines converge. And this quite often happens with a lot of these characters. As that body size gets smaller, apes and humans become less different. And that's an important point. And here we see, this is at the foot, bones from the foot, and the light, the ellipse with the light symbols is for great apes, and the ellipse with the dark symbols is for Homo sapiens. And you see that LB1 falls within the great ape distribution. So here we have Homo floresiensis once again looking more ape-like than hominid-like. One question I would throw out is, if, as it seems to be the case, the wrist was ape-like in the Hobbit, how did it make those stone tools? And so I've been looking for an alternative explanation, and following a number of other people, including Teuku Jakob, the late Teuku Jakob from Indonesia. I think that maybe this is a pathological condition called microcephaly. And it's important to note that there are 400 different genes that can produce microcephaly, and there are dozens of different conditions, and so it's not a single syndrome. There are many different syndromes. And if we look at the data, histogram on the left shows you the average brain size in modern human microcephalics, females at the top, males at the bottom, and the average is 400 cubic centimeters, exactly as in the Hobbit. The top right histogram shows us the age distribution. And there's an important point about this. The Hobbit is adult. And so if we're looking at microcephalics, we should look at microcephalics who survived into adulthood. And you'll see most microcephalics die before reaching the age of 21. And probably about a quarter of them survive beyond that. And in fact, the extreme case on that diagram is a guy who was 73 years old with a brain a third the size of mine and did pretty well. And these are relatively mild forms of microcephaly with this tiny brain and people who managed to get by pretty well. And so I think it's quite possible that the Hobbit was in fact a microcephalic with a mild condition of that kind. And there are also dental anomalies shown by the red arrow. We know that microcephalic syndromes go along with dental aberrations, and they go along with alterations of the skeleton below the neck. Unfortunately, I've been unable to find a single postcranial skeleton of a human microcephalic in museum collections, and neither has anybody else. And so we cannot say whether the postcranial features we see in the Hobbit are similar or not similar to a modern human microcephalic. Nobody knows. I'd just like to finish off with one. This is a recent study co-authored by John Terrell, who's a colleague of mine at the Field Museum, and it shows with dates the probable migration of modern humans into this region, Southeast Asia. And you probably can't see the individual figures. Let me just tell you that most of them are around 45,000 years ago. And the interesting thing is that very soon after Homo sapiens gets to India, Homo sapiens is in the mainland in Southeast Asia and then in those islands, and then is in Australia at about the same time. So when Homo sapiens got to the region, we went through there like a dose of salts. And I simply do not believe that modern humans didn't get to Flores 45,000 years ago. That's it. Thank you.
Thanks a lot, Bob. We appreciate you and Fred both coming here and engaging in this dialogue with us. And I'd like to get the actual dialogue part of the dialogue started now. And I'd like to invite those of you who are watching this webcast out there to join in with us and send in, following the instructions on the BU Anthropology Department webpage, subpage for the dialogues. I'd like to invite all of you to send in questions or comments that you'd like to address to either Dr. Martin or Dr. Smith or both. And if you would like, please come and help us continue this dialogue at 4 o'clock this afternoon in the Castle on Bay State Road, where we'll be having a roundtable discussion involving Drs. Smith and Martin and the BU faculty in discussion, and then a reception afterwards. So join us if you can. And now I'd like to begin by asking Fred if you've heard anything from Bob during the course of the last 24 hours, the last day that we've spent together, that causes you to rethink any of the things that you knew or thought you knew about the Liangbua material.
The one thing, Bob, I mean, obviously, from my standpoint, the real burr in the saddle is that small brain size. I mean, I think we both agree that this is a problem. How do you explain a human with that small a brain? I think that although I was vaguely aware of the fact that there are not very many pathological individuals where we have their postcranial skeleton with whatever the cause of microcephaly might be. That is certainly, I think, a cause for some concern for me if there hasn't been a comparison of the Liangbua postcranial remains with postcranial remains from individuals who are clearly microcephalic. Then how can we rule out the possibility that perhaps these bones might be the result of the same syndrome that caused microcephaly? So I mean that you hammered that point home very well for me, and I think that your question with respect to that is a very, very important question that should be answered before we make too many diagnostic statements that such is not possible, because as you say, we don't know that it's not possible because we don't have the data.
Bob, let me turn that around and address the opposite question to you in the other direction. What have you heard that struck you as new or thought-provoking or caused you to re-examine some of the things you thought?
Yeah, one thing I'd like to emphasize that comes from our earlier discussion today, and I pushed my case very hard in my presentation, but there are worrying things from my point. Worrying as far as the microcephaly argument is concerned. I see no reason to depart from the argument that the tiny brain is a problem. That remains a problem, and nobody has effectively explained that. But if it's due to microcephaly, it's worrying that in both mandibles, that we have both lower jaws, there is no chin. And the emergence of this chin here is a diagnostic feature of Homo sapiens, of our species. It's not present in any other hominid, but it's usually present in modern humans. And there is no chin and no sign of a chin either in the LB1 lower jaw or in its companion. And while I would be willing to believe that there was a single microcephalic hominid on Flores, I think it's unlikely that there were two. And the two mandibles are also separated in the layers, so they're maybe a couple of thousand years apart. And so We cannot explain that lower jaw through microcephaly, and it lacks a chin. But I do have individuals in everything. Microcephalics are very variable because there are so many different genes involved. Some of them have poorly developed chins, but I haven't found anything quite as primitive as you find in the two lower jaws. Talking with Fred really made me think back to that and recognize that we do have a problem there.
I have some questions that have been sent in by people out there in television land, and I'd like to throw them out on the table here for discussion. As I said, we'll be continuing this dialogue at 4 o'clock in the castle, so please come and join us then if you have contributions of your own that you'd like to make in person. So one of the first questions I've gotten here is the following. Apart from the obvious small size, I'll direct this to Bob, does the virtual endocast reveal anything else unusual about the physical structure of the brain?
Yes, and in fact, this is what has been emphasized. Dean Falk and her colleagues have done a number of studies on the shape of the endocast, and argued in the original paper that the Hobbit actually shows some similarities, despite its small size, shows some similarities to Homo erectus. There are 2 points I would make. One is that shape to me is not important, and it doesn't explain the size difference, which is the key issue. So talking about shape does not resolve that issue. The second thing is that it's very difficult to allow for that great difference in size. I mean, if you're comparing a Hobbit brain, which is a third of the size of modern humans, and not much less compared to Homo erectus. There's a whole question of scaling and working out whether that, what happens to the brain if you just scale it down in terms of shape. And so we're kind of in unknown territory there. What I would say is that there are microcephalic brains that look remarkably similar to the Hobbit brain. The brains of microcephalics are incredibly variable, like everything else, but some of them actually look quite similar to the Hobbit brain in shape, and so there is not a clear distinction.
May I interject something into
You betcha.
this? Bob, I agree with you totally. Again, going back to the size of the brain, one of the arguments that Dean Falk has made, and particularly in this latest paper in Journal of Human Evolution, is that perhaps it's not so much size, but it is a reorganizational issue within even a smaller brain size that's important here. And she lays out 7 features that she feels are advanced in the virtual endocast, and argues that what we have here is a smaller brain that's somehow being selected for because of sort of expensive tissue kind of hypothesis things in an environment like Flores, which does have a low carrying capacity and is difficult. So you said before that we're treading into new territory. Might this not be an aspect of that new territory where it would be possible for something with this small a brain perhaps to be modern?
Let me come back to my first slide, because the key point is that Flores falls off the graph for all other hominids.
Right.
So if we're going to suggest that the Flores brain was reorganized, it's the only one that did it. Everything else follows the trend,
right,
and it gives us no surprises, and we have this surprisingly small brain. And I've been at a conference where somebody stood up and said, maybe the Hobbit brain was organized in a much more efficient way, so you could do most things like making complex stone tools with a third of a normal size. Mm-hmm. Why would this happen on this tiny little spot of land in the middle of Southeast Asia and nowhere else in the world?
Evolution is a chance process. I mean, just because something doesn't happen everywhere doesn't preclude the possibility, I didn't say the definitiveness of it happening in a prescribed set of situations in one place.
You know what I think of in this connection is bird brains. The smartest birds are as smart as the great apes in a lot of respects, tool making, linguistic ability, and so on. Brains the size of a walnut. Those are definitely, the differences there are definitely reflecting differences in organization, but they're differences that are 150 million years old. They're very anciently divergent lines, one of which, the whole bird radiation, has been under tremendous selective pressure to reduce weight so you can fly. And
but the birds are a good example because that's parallel evolution. Expansion of the bird brain was quite independent from expansion of the mammal brain. And a few bird groups have developed particularly large brains, notably the toucan group, which are fruit-eating birds, and the corvids, the crows and ravens and so on. And if you look at their brain in relation to body size, it's about the same as in a monkey. And so in parallel, they've reached the same level of development, whereas if you look at Homo floresiensis, it falls below that level.
Fred, let me ask you a question that's been sent in over our Facebook page. It seems that the recovery of genetic material would help settle this dispute. What are the chances of this happening?
My understanding is that they've tried to recover genetic material from the Liangbua specimen with no success. The fact about the matter is that things have to be just absolutely right to be able to preserve organic material that contains DNA in bones even as recently as 18,000 years ago. So the fact that we are apparently not able to isolate DNA from the Liangbua specimen is disappointing, but it's not particularly surprising because we have a lot of specimens in the fossil record that are within the last 40,000 years where people have tried to get DNA from them and failed. I mean, the failures are much more frequent than the successes. And so, although it's a disappointment, and it would be great if we could get it, that would explain a lot. For example, if it is a completely separate lineage,
dating back 1,800,000 years,
it should be very different. One would suspect, if this is a different lineage. But as far as I know, and again, I don't necessarily have all the information on this, but I'm certain they have tried without success to get DNA from the specimen.
Matt, that's a wonderful question, and I've heard through the grapevine that a group, I think from the Max Planck Institute in Leipzig, did try to get DNA. And there was a political problem about that, but nothing has ever emerged. There is no publication saying we tried to get a DNA and failed. What I do know is that Svante Pääberg's group in Leipzig has a way of telling whether you're likely to get good DNA or not from a skull. And I've seen no reference to them applying that test. Second thing is, this skull is 18,000 years old. They've been extracting usable DNA from about 20 or more Neanderthals dating back to 45,000 plus. And quite often the best place to get DNA is from the pulp cavity of a tooth because it's most protected there. And that was what I would do if I owned that skull, is I would remove a molar tooth and get into the pulp cavity if I really wanted an answer to this. And that's one way to go. This is one way in which we could get totally new evidence and perhaps resolve.
And it would be very important. I just would like to point out that just because we can get DNA from some specimens that are 45,000 years old doesn't mean we can get DNA from every specimen in that.
But I don't think the possibilities have been exhausted.
Probably
That's
not.
my point.
Okay. I'd like to wrap things up for this initial hour of today's first introductory dialogue by asking you both another question that was sent in over our Facebook event page. And this says that both of you in your brief written statements that you sent in advance, for which we thank you, and in your discussions today, have discussed the need for further information to settle the species versus pathology debate. So I'd like to ask each of you, what evidence would you need to be convinced of one position or the other? And I'll start with you, Fred.
A couple of more skulls that look like Liangbua, or don't look like Liangbua. In other words, I think Bob and I would agree, if there are 2 or 3 of these possible microcephalics in this population and nothing else, I think both of us would have a hard time explaining that as simply the result of pathology. So that would be the simple answer to the question.
Mm-hmm.
I completely agree with Fred. If we could get more material and certainly more skulls, that would really help. And if they find a second skull that's exactly like that with a similar postcranial skeleton, I will go back and totally rethink what I've been saying. My real point is that we have very limited evidence and we shouldn't be going so far on the basis of such limited evidence. But I would also like to say that I've tried to get access to the specimens and I've been blocked several times. And I requested CAT scans because I think there's a way of telling with the existing skull whether that is a microcephalic or not. And I was refused access. And I think this is just unacceptable. Science should not be operated in this way. Molecular biologists don't do this. Why should we get away with this?
Well, and I certainly agree with you that in order for science to operate properly, different research groups with different points of view have to be granted access to the material. It's one thing for a discoverer to have some time to be able to get their initial work out. I mean, they do have some proprietary rights to initial publications and so forth. But you know, when this extends over to 4 or 5 years and there is still no willingness to share full amounts of information with other scientists, I would strongly second exactly what Bob said. This is unacceptable. This is not what science should be about, and in that kind of a circumstance, we cannot have science because there is no replication of the studies on the information.
Let me ask you one final question, Fred. What would you need to know in order to be convinced of the proposition that the Liangbua 1 skeleton was not, in fact, a representative of a distinctive local population different from modern humans?
So in other words, what would I need?
What would you need to rule out that it's a different species hypothesis?
I'm going to sound like a broken record, Matt.
More skulls.
I want more specimens.
Right.
I think anytime you're dealing with one individual and there are suspicions about the normalcy of that one individual, that you're not going to get completely satisfactory answers until you're able to see whether or not this is the normal morphology for this group or not. Another thing that I would really like to see is a bit more evidence of this initial out-of-Africa radiation. I mentioned
you mean the early
pre-erectus that the new interpretation that this is the result of. I'd like to see some more evidence of that. There were arguments in Java, of course, about Meganthropus. You both will remember this, that Meganthropus was some sort of an Australopithecine-like thing. That, I think, argument has been pretty well shot down, but it would be more convincing to me if we had more of that evidence. Maybe the DeSilva genetic material is an indication of that, but again, I think just that one genetic sequence opens up a lot of problems as well.
Okay, thank you, Fred, and thank you, Bob, both of you, and thank you, all of you out there who have participated in and helped carry off and make successful this experiment in this new form of conferencing that we're inaugurating through the Boston University Dialogues in Biological Anthropology. Again, I'd like to invite all of you who have been tuning into this webcast to come and join us in an hour from now at 4 o'clock at the Castle on Bay State Road on the Boston University campus, where we will be continuing this dialogue and these discussions in a 4-person panel discussion involving Fred Smith from the Department of Sociology and Anthropology at Illinois State University, Robert Martin from the Field Museum of Natural History in Chicago, and a couple of local BU faculty with overlapping and corresponding interests who will continue this discussion on the meaning of the enigmatic little woman from Liangbua Cave. Thanks.